All right, this slide just shows you where you see substitution and elimination reactions in living organisms. These reactions actually are occurring billions of times a second inside of our bodies. Okay, both substitutions occur in our biological pathways, so SN1s and SN2s. Mostly, if you have to put on a methyl group. So here's an example where they take norepinephrine and SAM, which is S-adenosyl methionine, and what happens is you methylate the norepinephrine to form adrenaline or epinephrine. Okay, so it kind of shows you kind of an SN2 type of a mechanism where your nucleophile is attacking your methyl group. Okay, and basically this whole thing is your leaving group. Okay, kind of interesting that whole thing is our leaving group through an SN2 reaction, and these are the two compounds that you make. All right, here's another example of an elimination reaction. All right, this is imidazole. These are found a lot on our amino acids as well as our DNA. So these are very common inside of the body. You have a hydrogen. We haven't talked about these types of thioesters, but these hydrogens are very acidic that are next door. And it's kind of eliminating. Eventually what happens is the pi bond pops down here and the OH leaves as water. It leaves as a water group. So you're basically going from an OH here. So imagine that as a halogen. Now, we don't have a lot of halogens in our body. We actually just have chloride ions for electron transport and things, but not for reactions like this. We do not have halogens as leaving groups. So we have OHs as leaving groups. Phosphates are leaving groups inside of our body. So it's kind of you're plucking off this hydrogen, you're eliminating the OH to kind of form a double bond right there. And that is it for this chapter. This will give you kind of a good introduction. I am going to do a little bit more in class, so be prepared to take more notes in class.